ATP synthase is a rotating membrane protein that synthesizes adenosine triphosphate ATP from ADP and inorganic phosphate. For this reaction to happen, there must exist an electrochemical gradient to be countered. To dissipate it, the protein pumps proton to the negative charged surface, and during this pumping, the coupled ATP synthesis reaction occurs. The subunits involved in the process force the protein to rotate, and we wonder if this movement affects the lipid membrane where ATP synthase is inserted. For that reason, we'll build up a system by mixing purified E. coli ATP synthase and electroformed giant unilamellar vesicles by the use of biobeads to remove the detergent and ensure the protein to be inserted within the membrane. To check if the system worked properly, we used confocal microscopy. As we said before, there must exist an electrochemical gradient to trigger the protein to dissipate it and create ATP. So we use bilinomycin, that is a transporter of potassium cations. It will charge positively the inner of the vesicles, forcing the process to occur. To follow the evolution of the system, we trapped a pH probe, pyranine, inside the vesicles. Pyranine emits more fluorescent as the pH goes up, so if the protein is working, it will dissipate the gradient by pumping protons, changing the pH, and this event will be observable, as it happened left side of the poster. To test the validity of the results, we also ran a control experiment in the absence of bilinomycin, right side of the poster, where almost no changes were seen. This told us that the system was working, so we tried to get more information with another microscopy technique. For that purpose, we changed to phase contrast microscopy to obtain data about the movements of the membrane. So we again triggered the formation of ATP by adding bilinomycin and take films at different times, 0, 4, 8, and 12 minutes. For each frame of each film, the counter of the vesicles were tracked using a PECRO-based algorithm developed by Mike Mell from Paco Monroy lab. The tracked counters gave us information about the membrane's movements, and decomposing them through a Fourier analysis, we could get its corresponding fluctuation spectra. As we see in the inactive case, bottom right, the spectra remains the same at different intervals of time. However, in the active case, bottom left of the poster, the spectra goes up as the time increases. These experimental spectra can be fitted into a theoretical expression in which physical parameters of the membrane appear. Membrane tension, sigma, related to the stretching deformations, and bending modulus, kappa, related to bending deformations. Once the spectra are fitted, these parameters can be obtained. As we film it at different times, we can get the revolution through time. In both cases, sigma remained almost the same, but kappa didn't. While in the inactive case, kappa stays more or less constant over time, in the active case, its value lowers drastically, indicating a membrane softening. In summary, we find that ATP synthesis rotates this, promoting large out-of-equilibrium deformations in lipid vesicles, and thus inducing an overall membrane softening. Therefore, the rotation of ATP synthesis promotes mechanically adapted membranes with a high bending compliance and able to support high local curvatures. Our results evidence a mechanical functionality of the ATP synthesis for biomembrane restructuring and shaping. Thank you very much for your attention.